Welcome to the first ever live streamed episode of Artificial Intelligence in Healthcare, the Intelligence Roundtable. In this episode, we will delve into the transformative work of Ibis Therapeutics, a trailblazer in the realm of AI-enabled drug discovery. This event proudly features Wayne Guida and Bill Glauser, key figures behind the innovative force of Ibis Therapeutics. Wayne, as Schrodinger Inc.'s first CEO, played a crucial role in its ascent to a $2 billion company, laying a foundation of excellence that he now extends to Ibis. Ibis Therapeutics stands out for its innovative approach to healthcare, employing advanced AI and computational techniques to streamline the drug development process. A key focus of our discussion will be on the role of antibody-drug conjugates, or ADCs, a revolutionary class of biopharmaceutical drugs designed as a targeted therapy for cancer and other diseases. Ibis's work with ADCs exemplifies their commitment to developing more effective and precisely targeted treatments, showcasing the significant potential of combining biotechnology with AI. A significant part of Ibis Therapeutics' success story is the collaborations with leading institutions such as AbbVie and Mayo Clinic. These partnerships not only amplify the impact of Ibis's innovations, but also pave the way for advanced drug discovery and clinical trial processes, demonstrating the power of collaborative endeavor in pushing the boundaries of modern medicine. Looking ahead, we'll share insights into the future directions of Ibis Therapeutics and the ongoing challenges it seeks to navigate. This session promises a wealth of knowledge for industry professionals, investors, and anyone interested in the cutting-edge intersection of AI and healthcare. Hi, everyone, and welcome to the first ever live streamed episode of Artificial Intelligence in Healthcare, the Intelligence Roundtable. I'm your host, Doug Nisanoff, Managing Director of Intelligence Ventures. For those of you that are new to Intelligence Ventures, we are a venture capital firm that is laser focused on investing in US-based startups operating within the intersection of artificial intelligence and healthcare. And today marks the 1-year anniversary since we formed. Since February of 2023, we have met with over 120 startups that match our investment thesis, which is why today's guests are so special to us. After a year of rigorously meeting founders and countless hours of due diligence, which, by the way, huge shout out to my colleagues for their fantastic work with this heavy lift, we have finally identified our first investment, Ibis Therapeutics. There are quite a few reasons why we are selecting them as our inaugural investment. Let's start by talking about the current state of AI and its applications in healthcare. It can be used to help physicians spend more time with their patients and less time charting via voice transcription, right? It can be used in medical imaging to help identify and diagnose disease. But one of the current use cases that we are the most bullish on are within drug discovery. In our last episode of our podcast with Dr. Gavin Hurst, we spent a great amount of time discussing the arduous process that is drug discovery and how AI has the potential to make drugs better, faster, and cheaper. While AI-enabled drug discovery has become wildly popularized since the launch of OpenAI's ChatGPT in 2022, I need to emphasize there is too much hype out there right now. And I'd like to say that 1 in every 4 startups that we meet with are playing in the AI-enabled drug discovery space. And while there are green pastures and plenty of room for new entrants, what we were specifically looking for were startup founders with exceptional backgrounds in the space that we wholeheartedly believed had a differentiated offering and the team to capture the market. And man, did we find just that with Ibis Therapeutics. I'd argue that there are very few people in the world that have been moving and shaking the space as predominantly as their founders. So before we bring on our guests, I'd like to share an exciting opportunity for accredited investors. We're offering our first special purpose vehicle through AngelList Syndicate. This means streamlined access to select high potential AI healthcare investments, comprehensive deep dive analyses, our first one's about 13 pages to give you an idea, and the chance to co-invest with industry leaders, all within a community that shapes the future of healthcare. If you're interested, I encourage you to apply to our syndicate on our website at intelligencevc.com or reach out to me, connect with me after the show. You all are in for a very special treat today. So let's go ahead and bring on our guests, Drs. Wayne Guida and Dr. Bill Glauser. Gentlemen, welcome to the show.
Thank you, Doug. It's great to be here.
Thank you, Doug.
We're so excited to have you both on. So let's go ahead and just get started by talking a little bit about, you know, what got you guys into the industry. Let's start with talking about Ibis Therapeutics.
Well, Doug, Ibis Therapeutics is a tech bio company as opposed to, say, a biotech, which means it exploits an AI-driven platform to sort of serve as an evergreen source of asset creation, both for ourselves and our collaborators, and future in-licensors as well. Ibis has really two missions that are very clear. One, given a particular drug, find the best patient cohort for it. But conversely, given a patient cohort, find the best drug for it. So in that sense, it's sort of the epitome of precision medicine. And you think about it for a second, isn't that what bench to bedside and bedside to bench is really about?
Yeah, absolutely. And, you guys have had some significant roles prior to starting Ibis Therapeutics. I believe you started at Schrodinger Inc. And you both had some roles where you contributed to its eventual growth to a $2 billion market cap. So, what inspired you to bring this expertise to Ibis Therapeutics?
Wayne started this rolling. Dr. Guida was the person who actually pivoted Schrodinger from more of an academic exercise into a full commercial exercise. Wayne?
Yeah, I was fortunate enough to have been the first CEO of Schrodinger. We were a small fledgling company. I think we had $1 million angel investment, and we were focused on bringing computational methods and AI to the drug discovery process.
That's super cool. And so I'm sure you've seen a lot of transformation since you started. I mean, when did you start with Schrodinger? And how has AI changed the drug discovery process? And what enhancements have you seen since then?
Well, I started in 1995. That must seem geological epochs ago in the field. And back then, Schrodinger was focused on two things. Number one, what happens when a drug hits its target? And I wanted to know everything about that during that event, just before and just after. And the thesis was, back in the 20th century, seems so long ago, the paradigm was one disease, one gene, one protein target, one drug, a veritable magic bullet. And Schrodinger thought if it could understand exactly what happened, kind of like astronomers at the moment of the Big Bang, they would know everything about the evolution of something. What was something similar to that? But Schrodinger had a laser focus on understanding things from the ground up. They wanted to make a movie, and as if you could peer through a nano microscope and actually see the drug swimming around in aqueous solution in water and bindings, looking at different and hitting its target, and how the target would bend and flex and the drug would flex and they would come together, almost sort of like a ritualistic thing that they would do, but using physics. And that's where it all started. And on the back of a greasy napkin, Professor Bill Goddard at Caltech laid out a 20-year plan to fight disease with code. And I thought to myself, well, this isn't possible, but it'll be a heck of a journey. And that's how I joined Schrodinger. And Wayne, I think you had a slightly different beginning with Schrodinger.
Yeah, I joined in the end of 1998, so not that far after you did. But I had been interested in computation for many, many years. I was part of, but not contributed. I didn't contribute to the revolution that came about when the Apple IIe. I want to talk about history, Bill. The Apple IIe made it to people's desktops, and I was very, very interested in what we call computational chemistry, applying computers, even at that level, if you will, to understanding the physics of how molecules behave. And we were, you may remember, Bill, back in the day, talking about the movie, right? Because that was how we believed and still believe that we're going to solve this problem of drug discovery and development in a more timely manner, for one thing. But you remember the movies were 3D movies, right? We had the stereo glasses, and we could point to the areas in the binding site of a protein that
Yes.
were responsible for the interactions with small molecules, namely drugs. But that was those were the good old days. But 1998 for me.
So what president was that?
Oh, goodness. What president was that? Reagan? No, it wasn't
Teddy Roosevelt, I think it was.
Yeah.
That's post Reagan, right? So it must have been Clinton, right?
Okay. So just a couple of years ago. And I mean, my other very important question is, do you still have that greasy napkin, or is that just long gone?
It's long gone, but I decided to join the company because he picked up the check. And, and that would be in a shrine right now when Schrodinger went public. And they reached at one point a valuation near $7 billion at the peak of the tech boom 2 years ago. And just a testament to the fact that Schrodinger did something that was very different from other companies before or after. And this is some of what Wayne and I took from them, which is they developed their software in the heat of battle, and they were the first company to say, let us look at assets and develop them while we're developing our platform at the same time. Most companies did a waterfall development system. They develop it, have 20 developers in a room for 3 years and say, voila. And the market would say, we didn't order this. But what Schrodinger did, what we understood was, no, you develop in real time with interactive feedback. In fact, I would embed inside major pharmaceutical companies under NDA and actually listen to their medchem project teams and understand anthropologically how what we did fit into what they did. And that's where we understood that 90% of what other companies were doing were just not assimilatable in the context of real world drug discovery development workflows. They were shiny toys, very interesting, but couldn't be assimilated. That's what Schrodinger did differently, and that's what we brought. But Schrodinger was obsessed with the 20th century idea of one disease, one gene, one target, one drug. Nothing wrong with that. It's been very successful. But, and I'll segue to Wayne here, Wayne had a different take on it. In fact, he inverted this idea.
Yeah, so we were at Schrodinger interested in being able to screen computationally a huge library of molecules. That way, that would point us to the very molecules that we could go into the laboratory and make. Not that we would have done that initially at Schrodinger. That's done now, but back in the day, we would have partnered with a pharmaceutical company for this synthetic medicinal chemistry. And that has come to fruition. So that's the one drug that you want to find from an ensemble of molecules that binds to a specific protein receptor and modulates that receptor in such a way that would be beneficial from a therapeutic point of view. But there are many, many, many other proteins in the cell, in the human body, that the drug could interact with. Those are side effects. And so we had the idea, and this is with Dr. Wesley Brooks now, this was post-Schrodinger, but we had been thinking about this for years. Could we, in fact, invert flip the switch, as Bill said, and take a single molecule that we've discovered that would have a therapeutic benefit, and then screen that against a whole bunch of proteins. And then that would tell us which other proteins that molecule was likely to interact with when it was taken as a drug. And we could tell in advance what the likely consequences would be, what the likely side effects would be.
Yeah.
Starting with screening.
Yeah. And it's
computational screening.
And I'm not sure if you guys are able to see the comments as folks are rolling on in.
They flashed up for a moment. So
yeah, but there's actually somebody out there who's saying, one target, one drug has not been successful in their opinion. What are your thoughts?
We agree.
Yeah.
Well, we started a company based on that premise.
Yeah, and this is a perfect segue. So what are the major innovations Ibis Therapeutics is bringing to the field of drug discovery?
Well, Doug, there's a first slide that we'll show that will sort of map out the proteome. And what we're doing is sort of fingerprinting where the drug has been. Imagine that there's a bad guy and he robbed banks and everywhere. The FBI will come in, they'll dust for fingerprints. Where has that person been? Where did they come from? Where else did they go? And the whole causal chain. We are doing something very similar in the realm of the human proteome, as Wayne outlined. The cell has over 22,000 proteins. Any given cell type typically expresses about 10,000 proteins at a time. That's what differentiates one cell type from another. So we can look at up to those 10,000 all at once. And Wayne said, if you have an asset, or assets plural, you can ask the question, where have they been within the cell? Which proteins did they touch? And if they touch them, how hard did they touch them? Was it a strong affinity, moderate, or not any at all? And you can imagine creating a heat map of maybe red, yellow, green, something like that. And if you have, 10,000 targets, you can have all of these colors down a row for each drug. However, and that's what we're doing with Mayo Clinic now for, just as Wayne said, to look at off-target effects. So if we're trying to hit, for example, a protein called EGFR, epithelial growth factor, we don't want to hit the other 19,000, 20,000 other proteins that have no benefit in hitting it therapeutically. We can actually see this when you look at the blue dots on the right. So when you're looking to dial in greater safety, given a drug or a class of drugs, you want them to hit the fewest number of off targets. Well, if you're only looking at 100, you can't look for more than that. Other companies like Schrodinger look at what are called canonical anti-targets. Those are well known, things like hERG channels in the heart or cytochrome P450 in the liver. But there are, as Wayne said, hundreds, thousands of unknown, somebody once said, the unknown unknowns. You don't know what you don't know, what you're hitting. This is sort of like, you hear dark matter, dark energy. In some sense, this is like the dark protein space. No one's interrogated it yet. That's what we're looking at now. So with safety, you want to look at as many proteins as you can as putative off targets and make sure you're not hitting them. But reverse that script. Within any given target, it can have many different mutations, particularly in oncology and in infectious disease. In fact, that's the problem in oncology and in infectious disease. Tumors mutate. So you give them drug A, they mutate, now they're resistant to that drug, and you go on an odyssey. So on the left, you see what we're doing with AbbVie is we're looking at a given target, one target, as opposed to Mayo, where we're looking at hundreds, thousands of targets. Here we're looking at one, but within that one target, we're looking at a myriad of possible mutations that occur before treatment, during treatment, and after treatment. In that case, it's the opposite. Here, we want our drug to hit as many of those variants, mutations as we can for efficacy. Let me review what we're doing differently. On the right, we're looking at a pan-proteomic, large-scale, zoom-out proteins as off-targets not to hit to dial in safety. Conversely, when it comes to efficacy, we're trying to hit as many variants of a given target to make sure, whether in infectious disease, oncology, they don't escape treatment.
Super interesting. I'm going to close out this slide for just a moment here. And this is a great point. And so let's kind of zoom out a little bit. For those of us who may not be so familiar, what is an antibody-drug conjugate? Or I'm going to refer to it as an ADC moving forward so we can avoid the tongue twister. And how is it being used in healthcare, and what can they do for patients?
Yeah, well, antibodies are part of the innate immune system. They're the things that detect first line of defense. They're made in B cells, and any sort of foreign fragments from cells they'll latch onto, or fragments from cells they'll latch onto, and they'll present, they'll destroy. They're sort of like the utility infielder of the innate immune system. They do a little bit of everything. But here's the thing. Antibodies have to be very specific, because if they weren't, they would attack your own proteins. So nature designed it so they have to be hyper selective so you don't get autoimmune disease like lupus and rheumatoid arthritis and ulcerative colitis, all of these bad things. So that's why antibodies are like super laser focused, targeted on a target. Well, Wayne was just talking about safety. Wayne was pointing out that in his experience, you want to avoid off targets. Well, that's what antibodies do, but they're big, they're proteins. they can knock on the door to get in the cell, but they're really not allowed in. So the benefit is they're ultra selective, but they can't get inside the cell. Now, small molecules are the reverse. They're not nearly as selective, but they can get into the cell. Inside the cell, they can hit any sort of targets they want. Now imagine if you wanted your cake and eat it too. Antibody-drug conjugates take a small molecule, link it to an antibody. So the antibody is sort of like this cruise missile. It hones in exactly where you want it, laser guided, but then it delivers its payload inside the cell. And generally it's cytotoxic, so you get a kill rate of almost 100% once it's inside. So you get the benefits of a large molecule and the benefits of a small molecule all in one. What's not to like about that?
Yeah, targeted, very, it's a massive market. But this is not something that's new. In fact, we've seen quite a few, some of the biggest biopharma companies out there are working on ADCs. So my question to you is, how does your approach to ADCs differ from traditional methods?
Bill, if I could just interject very quickly. Yes, it's not new. One of my former students who unfortunately passed away at a very early age due to medullary thyroid cancer had worked many years ago, actually, in this field, the antibody-drug conjugate field. So, but it's only recently come to fruition.
Fascinating. And so, how are you guys, differing from what's out there right now?
Well, Doug, in a slide that we can show, I think they always say a picture's worth 1,000 words. And in this case, almost looks like, are we seeing the same thing? It looks like a musical note on this screen and not an antibody. What we basically do is we take a look at a actual target of an antibody. So all cells have this membrane around them that protects them, keeps the stuff in, good stuff in, bad stuff out, and maintains homeostasis in there in terms of its salt concentration, etc. Its balance. Now, it only lets certain things in. It gets messages from other cells, and they communicate with each other through these receptors on their surface. So if you would look at a cell under a microscope, it would kind of look like a golf ball in a way, with these little indentations on it, something like that. And in those indentations, that little golf ball, which is a cell surface, individual things can bind to it, which very often are antibodies or messengers. So when they create drugs that try to hit these targets, that's what they're aiming at. Now, what's interesting in these targets have half of their head outside the cell and their tails inside the cell. What they noticed 3-4 years ago was, if there's a mutation in the tail part, one end, one teeny little single nucleotide, one little amino acid out of 1,200 gets mutated, that effect travels all the way up through the membrane to the part that's sticking out and changes its binding. So pharmaceutical companies noticed that ADC targets are usually overexpressed in cancer. And that's how you spare normal cells from disease cells. Normal cells only express a few of these targets. Tumor cells express thousands, tens of thousands, hundreds of thousands. So if you give a drug that binds to that target, chances are it's going to hit the tumor cell, not hit the healthy cell. Just a matter of numbers, very simple. But they found that. They didn't understand it. If there was a mutation in that target on the cell, sort of the latch that the drug binds onto to get into the cell, one little mutation, even if it didn't overexpress, that drug was still very, very active to kill the tumor cells. They didn't understand why. And that's when AbbVie came to Ibis and said, can you explain and then predict when these mutations will cause this effect? And what am I talking about? Well, and if you show the next slide, this is a fingerprint of what we've had. And we've been discussing fingerprinting, a heat map. This is an abbreviated version. In the real fingerprint, it can have many more rows and many more rows and many more columns. It would be much more difficult, but we've simplified it. In this case, we're looking at a fingerprint of a target, a region of a target. We're looking at 5 different patient cohorts here. Patient 1, 2, normal, 3, 4. 1 through 4 may be diseased, and then the wild type is normal. We're looking at the color codings, and this is how this target reacts. And this is what Ibis pioneered, that we can figure out how these different sites on this target that's sticking out of the cell will change in response to any patient cohort variant. And notice how they cleanly segregate with each other. You can see they're all green mostly, they all red mostly, combinations of red mostly, red with a little black, a little green. You get the point. They're showing different patterns. This is generated using basic physics, things that Wayne and I did at Schrodinger, basic physics, but it's interpreted through the lens of artificial intelligence, machine learning. So what makes what we do different is that we seamlessly combine bottom-up, fundamental, physics-based, looking at a molecular movie, how things actually work in a wiggle and bind together. But at the same time, we take that as raw data and then we massage it using and train machine learning algorithms for patterns. In this case, this is a pattern for efficacy. So in this sort of stylized example, the two patient cohorts 3 and 4 would be more susceptible to the drug that, say, a given pharmaceutical company would already have in phase 2, phase 3, or already approved. The one in the middle is a reference, and then patient cohorts 1 and 2 would not be included in follow-up clinical trials or follow-up indication because they wouldn't be predicted to be susceptible. We've had 7 projects to date where we've had to make predictions like this, and in each case, our predictions have been borne out by experiment.
That is incredible. And this is a perfect, segue into my next question, which is, can you share some insights in your collaborations with institutions like AbbVie and Mayo Clinic?
Yes. Wayne, why don't you, with Mayo, I think you had started the ball rolling about safety. So it would be of interest to hear what we've done with Mayo as a safety perspective.
Yeah, so with Mayo, we're interested in a particular set of enzymes. They're related to each other. And there are a number of marketed drugs. These are called JAKs for short. And they're related to each other. And so it turns out that there are known side effects for these marketed drugs. But no one really knew, or before we started working, what the cause of those side effects was. So we used what we call molecular fingerprinting, which is this combination of one molecule screened computationally using physics-based methods against a ensemble of proteins, or the other way around, a ensemble of molecules screened against a single protein. So if you combine those, you think two concepts, you end up with a whole matrix of data that includes each molecule and how it interacts with every protein in the human genome. We're building that database as we speak. We have over 10,000 proteins now in the database. So it's database intensive, but these days, of course, we can handle large databases and apply machine learning to that data. And so what we were able to do with Mayo is a couple of things. One is to take these known JAKs along with known drugs and determine what the off-target effects might be with respect to the very proteins that are being engaged. And one of the things, obviously, we don't have time to get into all the details of the protocols and things like that, but turns out that thrombosis is one of the side effects. And when we first ran the program to generate the molecular fingerprint, we were hitting molecules computationally. Now, this is using physics-based methods. We were hitting proteins that would be involved in thrombosis. So we have done that now many times with many different projects and validated this molecular fingerprinting project and protocol. So yeah, that's the thrust of the Mayo project.
That's excellent. And actually, great question from Brian Silverman here. He said, so based on what you shared, biomarkers can then be utilized to recruit subjects in a more exacting manner as part of the inclusion-exclusion criteria.
Brian, that's exactly right. And that's exactly how it can be used both for inclusion and exclusion. And in the example I showed before with the ADC, those patient cohorts mostly in green would be included, but those in red would be consciously excluded. So you don't waste time. And by excluding patient cohorts, which can be sometimes just as impactful, you increase your effect size during a clinical trial, which is often diluted by patients that would have no chance of responding. But that's exactly right. And this is the quintessence of precision medicine. The difference is we work in conjunction with the liquid biopsies that AbbVie and others will do. And in quasi-real time, then we can analyze it and turn it over into actionable results.
Excellent. We have another great question from one of our advisory board members, actually, Dr. Gavin Hurst. He says, was there a common theme in the proteins that were hit by JAK inhibitors?
Yes, we did hit some enzymes that are involved in the blood clotting pathway, for example. The other common theme was, of course, other kinases, the Janus kinase. That's what JAK stands for. So that helps us to validate as well the methodology. When we are hitting targets that are sort of obvious targets, which you don't really need the computation for, that validates the whole process for us. So yes, we were hitting common targets, other kinase inhibitors that would be expected to interact with the JAK proteins, and then in addition to that, some others that weren't so obvious that would potentially be involved in thrombosis. And then a third tranche, which were not obvious at all. And that would be followed up eventually by testing those specific proteins with the JAK inhibitors.
Awesome. Thank you. One more from the comment section. I wasn't expecting such wonderful questions and thoughts here, and then we'll move on to some other stuff. But Dr. Frommo says, do the computational tools exist now to predict how individual mutations impact the structural alterations in a given protein and the potential biological impacts?
Yeah, Robert, that's a great question. And we do use AlphaFold in conjunction with cryo-EM studies as starting structures. Presumably, that's part of what AlphaFold could do. But in reality, and Schrodinger published a white paper recently on this subject, it needs what the Beatles used to call a little help from his friends. So it's not quite as simple as that. So what we do, we actually use a dynamical molecular dynamics. And so we animate it. We actually show the dynamical changes. So we look at both static structural changes, which are occasioned. And what really struck us was that when you make a single point mutation through a membrane 30, 40 angstroms away, how that signal, if you want to think of it, can propagate. To some extent, a small mutation in an amino acid, and sometimes something like a isoleucine to even a leucine, can occasion large dynamical changes very distal from that site. And analogously, a mutation can be analogized to a drug binding to that site. They have very similar properties. That was another key finding from this Ibis collaboration and what we do. So the answer is yes. We look at the static structural implications, and there are those. And it takes, as you can imagine, a lot of CPU time to do this. And one of the reasons why we're here, and one of the reasons we'll get to this later, in terms of what we would do with additional funding, would be to scale this up so our throughput and turnaround would be increased to do this in space and time. But most importantly, Robert, we can look at dynamical signatures, and what you were looking at in that heat map was not as much structural, but a dynamical signature.
You know, Bill, all those years ago at Schrodinger, when we were working on physics-based methods and polarizable force fields and things like that, those are methods now that are in our armamentarium that allow us to do these kinds of things. Also, the computers have gotten faster. We actually use graphics processors to run these simulations. We call them molecular dynamic simulations. But believe it or not, graphics processors are ideal for that type of computation. So we string together a bunch of graphics processors and away we go.
Doug, Wayne brings up a great point. Ibis sits at the center of a web. When you think about all these technological innovations like AlphaFold, AI for proteins, and you think about quantum computing that's around the corner, and you think about, as Wayne was talking about, GPUs, graphical processing units, any sort of benefit, we have an unfair advantage that we somehow non-linearly, exponentially improve as these external inputs. So a rising tide in a disproportionate way lifts our ship.
Yeah, absolutely. And this is another thing I wasn't even planning on discussing it, but I love the direction you're heading here, which is just quantum computing. Where do you see that going in the future? And I'm super excited about it, and I see the potential. But many are arguing it is not there yet. It is a large, expensive toy that can be there someday. I like to think of it as computers back in the 1950s, perhaps, where you have this very large room and some kind of geeky guy goes in and says, hey, check out my toy. And everyone says, this has no use, but okay, very nice. How much did that cost you? But where it's today, do you think that quantum computing is heading in that direction as well?
Bill, I'll let you talk, but I can't resist talking about the Cray computers that I used back in the 1980s that took up a whole room, right? And the graphics processors that we use today are, my thumb, the size of my thumb, basically, and with thousands, if not hundreds of thousands times the power, right? But yeah, sorry, go ahead, Bill. I couldn't resist that.
Wayne, you bring up a great point because, computers back then were special purpose. They were coded, hard coded. You really didn't have software per se. And that's where quantum computers are today. So when you think about it, it's not just the hardware that's a problem, freezing something to absolute zero so the qubits don't dephase from 0 to 1, but also the software. So this will proceed the same way. So the question really is, the right question is, what will be the first and second and third killer application, special purpose software for this new hardware? That's the question.
And it will happen. I mean, we've seen it happen so many times before in the computer field. We're old enough to remember the Cray computers and the Apple IIes and all of that, and it will happen.
So when is the question? When do you speculate?
Bill, I'll let you take that one.
I'll let the other Bill. Bill Gates famously said, we tend to overestimate what we can do in 2 years, but underestimate what we can do in 10 years. And, if you follow Peter Diamandis and the singularity universe about exponential growth of things, you only know after it happened, that sort of inflection point that it reaches, that hockey stick. No one can predict before, but once it happens, it accelerates quickly. And we see this in biology, and we see this in so many fields like CRISPR technology and next generation sequencing and multi-omics. And we see, and the same thing with ChatGPT-4. So ChatGPT-5 will not be 2 times, 3 times, but it will be 10 times, and then the next will be 100 times. This is the exponential universe that we live in today. Quantum computing will be no different. The first application obviously will be NSA, DoD, cryptography. They always get first cracks at everything. It was the same thing with computing. Wayne, in our day, DARPA, Defense Advanced Projects Agency, always had first crack at all the new computers. But in terms of the killer application, Doug, to answer your question, you've got to imagine that protein structure prediction is going to float to the top of the queue because that problem maps so nicely onto some of these architectures. And also, think about it, if you're developing CPUs, if you're NVIDIA or Intel, you want to partner with companies and develop applications, chipsets for companies that are going to chew up ungodly amounts of CPU power. Well, this is where it's at. People thought it was genomics and genomic sequencing, and that was the Craig Venter in the race, and that was going to unlock it. But what they didn't realize was proteins are the actors on the stage. It's not genomics, it's proteomics, and that's what IBIS does. And so to answer your question, probably the NSA will get quantum computing first. Sorry about that. But shortly thereafter, we'll be close to the top of the queue.
I have no doubt about that. Thank you. Super insightful. And, I'm reading the comments now. Some people are, I see somebody agreeing, saying, like a computer in every household didn't happen until a decade later than predicted. And now look at where it's gone. And, I think we can say the same thing about modern medicine, which is we are in the very early days of drug discovery and revolutionizing how we think about treating disease. And I think in, let's call it 20, 50 years, if you look at a graph of all the drugs that will be discovered and what humanity is going to look like from a drug discovery standpoint, we are going, right now, it's just the very tiny bottom of the graph before you reach, the amount of drugs and knowledge we are going to accumulate. So this is the time for that. One of the things I want to talk to you and then just pivot back is to, safety and efficacy in drug discovery, in drug development. Can you guys discuss the importance of safety in drug development and how your platform is addressing this?
Well, I'll leave this to Wayne, but let me just quickly toot his horn. Most tech bio companies are started by people in the tech field, and biopharma companies are people with, drug discovery experience. To de-risk everything we're doing, Professor Wayne Guida, who over 3-4 decades has seen it, been there, done it, and basically his whole journey, and I'll let Wayne speak for himself, but he's too modest to say all this, is that we're unique in that we have a CEO of a company that has actually discovered drugs in multiple contexts and yet is at the forefront of technology. I don't know other companies that have a person in this critical position with this background who spent a career balancing both efficacy and safety. Wayne, all yours.
So I'll tell a story that Dr. Wesley Brooks at the Moffitt Cancer Center and I, a number of years ago, had been thinking about this whole process of, is there a way to get at safety while you're in the early drug discovery process so that you can build into your molecule the selectivity you would need to hit the target that you want it to hit, but not hit the multitude of targets that it's likely to hit. And we were working with a group, actually it was a proteomics group at Moffitt, who was also, of course, interested in this as well. But they had the technology to determine whether a specific molecule was hitting a specific protein. And so Dr. Brooks and I, sort of scratched our head and said, well, why not? Why can't we take a single molecule and allow it to interact computationally with a whole plethora of proteins? We had been doing screening of a bunch of molecules, hundreds of thousands of molecules on the computer, whereas if you do this physically, you can ultimately screen maybe 100,000, but it's going to take you many days to do it, even with the most modern equipment. But we've screened actually millions and millions of compounds in a few days doing it computationally. So to make a long story short, we put together this algorithm. I think the first one, Dr. Brooks' program, wrote the code, and we called it virtual target screening because that's what we're doing. We're screening for targets, which protein targets are also engaging with the small molecule that we ultimately want to put in patients, right? You really want to know that, right, as early as possible. So that's how we got into this whole area. And then, Bill, it was you, quite frankly, who said, well, let's combine the two, right? Combine the small molecule against an ensemble of protein targets with the with the group of molecules against each and one of those targets individually. And that has led us now to what we call molecular fingerprinting, which is you saw a small panel, if you will, of the individual data. But that can be we have on the order of about 10,000 protein structures at this point, which is representative, very representative of the entire human proteome. But we have access to millions of small molecules that we can dock against. We can allow to interact with each of those proteins. So we have data, quite a bit of data that can be handled. We have data scientists who have worked with us to make that process possible. But then we also have machine learning that can take that data and pick out the nuggets from what's in the data. So that's pretty much where we are. That's what we call molecular fingerprinting.
And
that's
so, and somebody's asking a great question. So if you think about all the data that you're feeding the AI ML, you know, they say it's garbage in, garbage out. It's only as good as the data that you trained it in.
Right.
The other thing that's been fascinating is, from a language standpoint, AI ML is a complete anomaly. I mean, you are able to take any language, put it into ChatGPT, and translate it almost instantaneously. Now, what would you say is the next language to be learned by ML? And somebody from our audience is asking a great question: Is protein crosstalk the next language to be learned by ML?
What a wonderful question. At Columbia, Barry Honig was looking into PPI and was interrogating this about 12 years ago, 15 years ago, hitting right on it. There is a language. And in fact, protein-protein, drug-protein interactions do define a language. They have a grammar. They have a syntax. So it can come out the same way. There are probably adjectives and adverbs and prepositions too. The answer is absolutely yes. There's a parsimonious set of rules, and it is it's not so much, it's complex. So it's nonlinear in a way, but then again, so is semantics is nonlinear with inflections and nuances. So I would say this question that Glenn raised is precisely the right question to ask. And we will reply affirmatively, yes.
And in some sense, Bill, there's nothing new under the sun as well, right? So a number of years ago, this is over 10 years ago now, we had a major grant at Moffitt to look at molecules that function by disrupting protein-protein interactions. And so it was a significant grant with multiple laboratories across different departments. But that's where one starts learning that language, right? It's from the structural information and the computational work that helps us understand how does a small molecule come into play that can disrupt a protein-protein interaction. That helps us tease out what the crosstalk is.
Yeah, and he responded, perhaps the emergent behavior that stems from that will be remarkable, which I agree.
I agree too.
Yeah, absolutely agree. So let's transition into the future for Ibis Therapeutics. So what are the next steps for your company in terms of research and development?
Well, right now we're working on a new project with our major pharmaceutical partner. And we described before that we think about a quadrant, and you think about discovery and development. And then on the other axis, small molecule, large molecule. What we described so far is small molecule discovery with Mayo Clinic, helping them with safety, and then large molecule clinical trials with AbbVie to help them with efficacy. Now we're moving into a third quadrant, which is small molecule clinical trial enablement. So you start to see that we're filling up these quadrants now in the generality. So the next steps will be to do a few things. Generalizability, to make it fill in all four quadrants. Small, large molecule discovery, small, large molecule clinical trial enablement, cover all four. But then secondly, automation. So we have the right throughput and the right turnaround time so that we can address multiple clients, a growing number of partners at the same time. But in order to scale, we need to automate certain things. And to automate certain things, we need to do a few things, one of which is to convert a lot of what we're doing into a data science problem. Much as we use machine learning to deconvolute the effects for safety and efficacy, we can also use machine learning then to actually look at anthropologically what we're actually doing in our work. And so to codify that so that we can automate more and more of what we do. It's interesting. There's always a certain amount that you want to leave to the human, that which humans do best. So we often look at AI as not as much artificial intelligence as augmented intelligence. So if we can automate the parts that are really programmatic and algorithmic and leave to humans what they do best, then with the assemblage of talent that we have and the algorithms that we have, 1 + 1 can equal 5. That may be poor math, but it's great synergy. So the next steps would be to automate, scale, and generalize. And that's part of what our interest in capital investment, that's the use of funds would be for. Internally, to expand the platform, as Wayne was saying, right now we cover a proteome of 10,000, but we'd like to go to the entire proteome if and when we can. No stone left unturned. And then we'd like to automate, scale, and generalize, and then to use that externally to be able to engage a growing number of partners. Fortunately, we started off with the third largest pharma in the world and the marquee biomedical research institution. My gosh, if you got to start somewhere to do a proof of concept and validation, well, you did pretty good in that way. And that's what we learned from Schrodinger. We learned that when you develop your algorithms and you build your team, do it in the heat of battle. You get instantaneous feedback that way.
Yeah. And, I had the pleasure of speaking with some of your partners. They left glowing reviews of the work that you did and the data and the outcomes that you were able to produce. So that speaks that speaks to your capabilities and to the power of what you guys are doing.
And one thing, we with regard to AI, a number of the AI algorithms will give you an answer, right? But you don't know why they gave you the answer. Because our technology is physics-based, and we use graphical user interfaces for this. So we have a spot on our matrix, if you will, in the panels that you saw that lights up green. So that protein is doing something with our drug. Well, now and we're building graphical user interfaces for this purpose. If we click on that panel or that part of the panel, that cell that lit up, then that can take us to the very protein, the pictures and movies that Bill talked about at the very beginning, and we can understand why that molecule is interacting with that protein. That helps us to design perhaps out of that molecule the ability to interact with a protein we don't want it to interact with, and thereby alleviate the side effect.
Yeah. Okay. So what are some of the challenges you foresee in the near future?
Well, the bar keeps getting raised. And so we look at the challenges that are out there for design of novel modalities. So this is not limited to just traditional small molecules like Tylenol, Xanax, things like that, but also things like PROTACs, things that bind two things at the same time, even peptides for that matter. So you can imagine ascending the chain of different modalities. And the nice thing about generalizability, as Wayne was saying, we call our AI causal AI. Most AI companies out there are peer companies. They do excellent work, but it is veritably a black box. And if garbage comes in, garbage will come out, but you won't know why. Our AI is it's explanatory, both predictively and retrospectively. So you can actually eyeball it and do a sanity check. And as Wayne said, you can actually design around the results. Not so with other AI methods. You'll never know why they go wrong. You've seen ChatGPT-4 go horribly askew, but nobody knows why. Here, you can then work backwards because it's a causality-based AI. So our biggest challenge today is to do what I said before, which is to scale this up. It's been validated by the third largest pharmaceutical company and the premier biomedical institution. And the next step is to scale this up so that we can have more impact and more leverage. But also what we want to do is to be able to more rapidly integrate with experiment, dry lab, wet lab. So we firmly believe, and some of the people who work with us actually have experience in the wet lab too. So we speak the language of experiment. Many people in AI are dry lab only. They're brilliant at what they do, but they don't interface well with the so-called real world. Professor Guida has worked in the real world. He's made compounds. He's worked inside major pharmaceutical companies over and over. And so therefore, we understand how to translate. So if you want to know why most of these companies have problems and the real challenges, it's integrating and interacting with the real world. We're blessed to have people, almost all of us, who have one foot in AI, computational physics, computational chemistry, quantum mechanics, and the other foot in experiment, whether it's cell biology, medicinal chemistry, physical chemistry. One of the few companies that spans both, and that has always been the biggest challenge. That iterative cycle of asking a question theoretically and answering it experimentally and turning that hypothesis crank ever faster with fewer resources per crank. That's the real challenge. And Doug, I would say we're addressing it by working on real world projects, clinical assets, as well as new to the world assets, because each time we use our platform to predict what a new round of assets should be made, it makes the assets better, but that feedback makes our platform better, which in turn makes the next round of assets better, which makes the platform, well, you get the point. It's an ascending spiral. And that's the challenge, to have the capital to continue to do that and build on that paradigm.
Yeah, absolutely. And I think that's a great place for us to, for my last question is, for those who are interested in learning more or supporting Ibis Therapeutics, what are the best ways to engage?
Well, first and foremost, tell your colleagues about what you've heard today. If you like what you've heard, if it stimulated you intellectually, if you've heard things maybe you hadn't heard before in this way, let somebody else know. Maybe they're outside your field, maybe they're not. Spread the word. If one tells two, two tells four, you get this exponential wave. And what it will do is it'll spread this idea, because it's an idea whose time has come. So first and foremost, I would say, discuss this with your colleagues. And by the way, the best way to learn a subject is to have to explain it to someone else. So if you want to retain what you've heard today, take the time today, tomorrow, the next day to encapsulate in any way, shape, or form. And I would also invite you to make this your own. Sure, we've come up with a lot of ideas. Yes, we've been in this for 35, 40 years, and we have a lot of expertise, but you also have expertise and you bring a new perspective to this. So I would invite you to take what you've heard today and sort of recast it in your own minds based on your own experience and your own applications, and make this idea your own as well. And I would invite you to do that. And you can join us through reading the literature, looking at our website. Doug and I, we will come up with deeper dives into many of these individual subjects, which will push out to you. And the opportunity to, as a scientist, of course, not a salesperson, I'm a scientist first and foremost, but the other way is the capital footprint through AngelList.
Thank you very much. Gentlemen, this has been phenomenal. Really enjoyed the conversation. Thank you again for taking the time.
It was our pleasure, Doug.
Thank you, Doug.
Great question. And thank you to the audience for some wonderful questions.
Yes, absolutely. And we're going to go ahead and we'll do some closing remarks here. So as we close out today's conversation, again, I want to thank our amazing guests, Wayne Guida and Bill Glauser from Ibis Therapeutics, for sharing their groundbreaking work and vision with us. It's clear that the path that they're paving in AI-enabled drug discovery has the potential to transform healthcare and patient outcomes significantly. That's why we are so bullish on it. So to our audience, thank you for joining us for this milestone episode. And I'm really grateful for the questions. Very thoughtful. I actually wasn't expecting how insightful you guys were. So thank you. Your engagement and your curiosity make events like this so rewarding. Remember, if you're an accredited investor interested in the transformative intersection of AI and healthcare, visit us at intelligencevc.com to explore how you can be a part of this exciting journey through our special purpose vehicle with AngelList Syndicate. Stay tuned for more deep dives into the AI healthcare landscape and keep fueling the conversation that drives innovation forward. This is Doug Nitsnoff, signing out.